Alright, hi everyone. Welcome to the last video for the chapter on molecular genetics. Right, so in this video, we'll explore what exactly are genes and what are some of their functions. And that's why we'll complete page 5 as well as page 6 of our notes. So, what exactly are genes and what exactly are their functions? Now, in the earlier video, right, I mentioned briefly towards the end what is the definition of gene. So, gene actually refers to a specific sequence of nucleotide within a DNA, and it controls the production of a specific polypeptide. In other words, it is a part of the DNA that allows us to have certain characteristics. So, as mentioned earlier on, this is a definition of a gene. Right? Do take note that in the definition of a gene, we need to specify that it's meant to control the production of a specific polypeptide. So each of the polypeptide has its own specific gene that allows it to be formed. Right? And to recap, right, polypeptide refers to a string of amino acids. Right? Because later on we'll talk about amino acids. And from polypeptide, you can use it to form proteins after that. So within a DNA molecule, right, there's something that we call a genetic code. Now, what exactly is a genetic code? If you recall earlier on, these are the basic units of DNA. They are called nucleotides. And within a DNA, there can be millions or in fact billions of nucleotides being arranged in each of the strands. Remember that the DNA is two anti-parallel polynucleotide strands, right? So within each strand, there's billions of nucleotides, and when you look at these nucleotides, right, they actually code for a certain piece of information. So the information that they code for is every 3 nucleotides actually codes for 1 amino acid. In other words, every 3 nucleotides give instruction for the cell to produce one type of amino acid in the entire chain of polypeptide. This is a bit similar to what we learned earlier on in Sec 2 or Sec 1 in ALP, where we learned about Arduino. Right, so in Arduino itself, there's also the idea of coding, right? So by keying in a certain set of instructions, you can ask the Arduino to do a certain type of function. Likewise, over here in the DNA, if we were to read the nucleotides based on a certain sequence, every 3 nucleotides will allow the DNA to know that it must code for a certain type of amino acid. So this is actually how the DNA codes for it. If you recall earlier on, there's 3 types of nucleotides. There's 4 types of nucleotides they will come across. There's those with the thymine, there's those with the adenine, there's those with the cytosine, and lastly, those with guanine. Right? These are the 4 types of bases. Depending on what bases the nucleotide have, we can actually categorize them according to what you see in this table. So remember, every 3 nucleotides code for 1 amino acid. Right, so this table is just meant to serve as a reference, right, to allow us to see that, for example, right, if there's TTT over here, in other words, meaning that there's a thymine nucleotide next to a thymine nucleotide next to a thymine nucleotide, so 3 of them, this will code for this type of amino acid that is given the short form as PHE, right? But there's no need for you to go and memorize this table because this will not be something that we want you guys to remember for the exam. It's more important to know that every 3 nucleotides code for 1 amino acid. So using the genetic code, right, what can happen is that the DNA can be used to make polypeptide. So now let's take a look at a video to understand how exactly is DNA being used to produce polypeptide by a 2-step process.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Right, so as you can see from the video, this two-step process firstly is what we call transcription. In transcription, it happens within the nucleus. So what happens is that DNA is being used to form what we call mRNA. Right, in other words, mRNA is also known as messenger ribonucleic acid. So from this messenger ribonucleic acid, that's where our cell, with the use of ribosomes, allow us to produce the polypeptide coming out in the strings. So what happens is that the DNA itself has a lot of nucleotides. Every 3 nucleotides codes for 1 amino acid. What happens is that the DNA will be transcribed into a single mRNA, which is then used to inform the ribosome on how to form the amino acids in order to create the polypeptide that we want. So, for example, let's say that this is the DNA strand, right? So all the alphabets over here represents one of the base. So T is for thymine, A is for adenine, C is for cytosine, and G is for guanine. So with this DNA, you will know that actually every three of the nucleotides actually codes for 1 amino acids. Right? What exactly is an amino acid? We don't need to care about them, right? That's only for us to know. But we can read the sequence as in every 3 is one set, every 3 is one set, every 3 is one set. Okay, so that is what you need to know about the genetic code. In transcription, this is used to form what we call an mRNA. Now, mRNA is a bit different from DNA in the sense that they don't have the alphabet T. I mean, instead they have this called uracil, which is U. So for mRNA, what happens is that the A will actually pair with U. Okay, so any form of base that has a will be paired with a uracil instead of A with T. Right, so this is the part that's different about the two. Then the next step, translation. In translation, what happens is that the ribosome reads the mRNA, right, interpret it as a code, then use it to form the strings of amino acids as you can see over here. Right, so then this will form your polypeptide. So this entire two process actually allows us to understand how exactly are proteins being synthesized. So we use the process called transcription and translation to describe protein synthesis. So this is just a diagram to illustrate what is happening. This is how DNA looks like. In the process of forming the mRNA, it goes through transcription. So that's where you get your mRNA molecule. Then when it exits the nucleus, when it reaches the ribosome, that's where you form your polypeptide by the process of translation. Now, what happens, right, when the sequence of nucleotides in the gene is changed? So right now I'm talking about this process called mutation. So you have learned about mutation in the earlier chapter in inheritance, but what is important over here is to take note that we are going to focus on the type called gene mutation. Right, so in gene mutation, what happens is that the nucleotide sequence actually changes. Remember what I said earlier? Gene is a sequence of nucleotide, so if you go and change one of it to something else, it will affect the amino acids that can be formed. In other words, it affects the type of polypeptide that can be formed. Not all mutations are harmful, right? So it really depends on the type of questions that you see. So two examples of gene mutation which we learned in the earlier chapter is albinism as well as sickle cell anemia. So just to recap, albinism is a result of a lack of a certain type of pigment, right, in the body. So as a result, the person usually have very fair skin. And this is a type of gene mutation. Sickle cell anemia, also a recap, right, is whereby a red blood cell doesn't look like the normal biconcave shape. It actually has a crescent, a moon-like structure that affects its ability to transport oxygen. Alright, and with that, we have come to the end of this entire chapter. Now, this is a pretty short chapter, so it's important to try out the practice questions within the Topical 10 years series so that you have a better understanding of what are the types of questions that you will come across.